Hi students, today we are going to learn a small concept from Molecular Basis of Inheritance chapter. The concept is processing of hnRNA. So this hnRNA is a precursor of mRNA produced. So what is RNA formation process called as transcription? So transcription will be following DNA replication. So first is the DNA replication process. So from DNA, the information contained is transferred or copied to RNA. So this process is known as transcription, and all types of RNA will be formed at the end of transcription. So prokaryotes and eukaryotic transcription differ in one important aspect is formation of hnRNA is there in eukaryotic organisms. In prokaryotes there is no hnRNA formation. What is this hnRNA? So HN stands for heterogeneous nuclear RNA, and this will be formed only in eukaryotic organisms. So what is hnRNA means? mRNA will be produced as a precursor in the form of hnRNA. Okay, so mRNA is not directly produced in case of eukaryotes. It will be produced as hnRNA. So these are the other names of hnRNA, precursor of mRNA or primary transcript. Okay, so in order to make hnRNA into the mature mRNA, we need to do processing of it. Okay, so these are all the steps of hnRNA processing. Right? So let us look at the structure of hnRNA first. Our 5' end and 3' end, and in this hnRNA, two types of sequences will be contained. One is Intron and other one is Exons. In Introns, the sequences are non-coding. They are just intervening sequences. They are not going to code for any proteins. But Exons are the coding sequences. So what happens in our mature RNA? We should not have interons, but exons should be present. So all this processing will be contained in the topic today. Okay. So what are all the steps in processing of this hnRNA? We will see it now. Okay. So the first step is capping. Okay. So cap is our 5' end. So in the hnRNA, our 5' end is our head cap. Okay, so in 5' end, an unusual nucleotide is added. Okay, the nucleotide is methyl guanosine triphosphate. Okay, so this methyl guanosine triphosphate is added at the 5' end of the hnRNA. This is the first step. Second step, if you see, it's known as tailing or polyadenylation. Polyadenylation means many adenylate residues we will be adding at the 3' end, that is the tail region of the hnRNA. So here around 200 to 300 adenylate residues will be added at the 3' end. Okay, and in this particular process, that is tailing process, we have to concentrate on one more content, that this polyadenylate addition do not need any template. Okay, so polyadenylation is done in a template independent manner. This itself is a separate MCQ based question, you will be getting it. Okay, and simultaneously the next process, splicing, will be happening. So we have added at the 5' end methyl guanosine triphosphate and at the 3' end polyadenylate. Okay, so now in order to become this hnRNA to mature mRNA, we have to remove the interons, that is non-coding sequences, and join the exons. Okay, so this exons will be joined in a defined manner, in an orderly manner they will be joined. Okay, so this process is only known as splicing. Right, so when all these three steps are completed, we will be obtaining our mature mRNA. Okay, so the fully processed mRNA will be formed only after processing of the hnRNA. Okay, right, so this mRNA will be transported from the nucleus to the cytoplasm after processing. Okay, so these are all the steps in case of processing of hnRNA. So how do I conclude that which is occurring first and which is next? So what is the sequential order of these steps means? This was asked actually in our NCERT previous year. In theoretical part of the textbook, it will not be mentioned, but if you look at the diagram in NCERT, transcription in eukaryotes diagram in NCERT, you can very well clearly understand that capping is followed by tailing, then completion of splicing will be there. Okay, so the order of the process is indirectly mentioned in our NCERT itself. So the diagrams, sequence of the diagrams, everything is crucial when you're learning NCERT. Thank you. All the best.